welcome everyone. Thanks for joining us today for the Tools and Technology Seminar Series. Today's session is remote only, just meaning that we do not have a live viewing opportunity in Palmer Commons. That's going to be true for next week too, at a minimum. After that, we'll just sort of have to get a sense of how things are going with COVID. If you have questions during today's presentation, you should feel free to put them in the chat box. We'll be monitoring that so that we can let our speaker know if any questions come in. So today's speaker is Jonathon LeFaive, and he is the Senior Applications Programmer and Analyst in the Center for Statistical Genetics. So thanks, Jonathon, for joining us today, and feel free to start your presentation when you're ready.
Thanks, Marcy, and thanks everyone for joining. As Marcy just said, my name is Jonathon LeFaive. I'm a staff programmer in the Gonzalo Abacasis lab here at the University of Michigan. Our research focuses mainly on the development of statistical tools that aid in studying genomic variation important in human disease. Currently, one of our main efforts has been contributing to the NHLBI Top Med initiative, acting as the project's informatics resource center. And in this role, we have analyzed many petabytes of whole genome sequence data. And today I'm going to talk about some analysis and storage approaches we have developed that are designed for genomic data sets of this scale. First, I want to show you an overview of how these data sets have grown in recent years. Notice that in addition to the number of genome sampled, we are also displaying the number of genomic variants detected. And this is to show that these data sets are growing on two dimensions. The more recent projects are employing high coverage whole genome sequencing, which is boosting the number of variants being detected within these large sample sets. And so with this technology, the more samples we sequence, the more variants we are detecting. And now a little background on the storage formats that have been used in recent years. The standard file format for storing genomic variation is the variant call format, or VCF. It's a text-based tab delimited format, and it's also coupled with a binary encoding called BCF. Looking at the toy example here at the bottom of the slide, these files contain a set of headers that describe the contents of the file, and then a table of detected variants. The first 9 columns contain site-specific information, and the remaining columns contain individual or sample-level data. So 1 column per sample and 1 row per variant. In this case, we are seeing a matrix of integer encoded alleles that an individual carries at a particular locus. So as whole genome sequencing data sets approach millions of samples and hundreds of millions of variants, these matrices are becoming quite large. But the key point I want to convey here before going forward is that the majority of these variants are extremely rare, resulting in these matrices being very sparse, or in other words, most of the values are 0. So to capitalize on this sparsity, we've developed a file format called sparse allele vectors, which I'll more commonly refer to as SAV. It's an extension of the BCF format I mentioned on the previous slide, and it produces smaller file sizes than BCF and is also faster when it comes to reading and writing files. We've also made some changes to the random access of data. So we're talking querying subsets of the data, either subsets of samples and subsets of variants. And then also to complement the file format, we've developed a full-featured C++ API and command line interface. And I have a link there to that software. All right, what I'm going to what I'm going to try to illustrate with the next few slides is how we exploit the sparsity of genotype data to compress the genotype matrix. And what we're looking at here is basically like a toy example of some genotype calls you might see in dense form. So first we discard the values that are 0, and then record the offsets of the non-zero values within each row. And then in order to improve downstream bit level compression, we actually store the relative offsets instead of the absolute offsets, since these contain repetitious integers. And even with this illustrative example, there's a notable reduction in what's being stored, but in reality, the large call sets are much more sparse and the reduction is far greater. There's also an optional method that can be applied in SAV for dense data types. This is the positional Burrows-Wheeler transform, which was originally described in a Richard Durbin 2014 paper. This sorts each row using a mapping that is based on the sorted order of the previous row. And so as shown here, the first row remains the same, but the entries in the second row are mapped based on their sorted positions in the first row. For the third row, they're mapped based on the second row, and so on and so forth. This results in sequences of repeated values, which is advantageous for bit level compression. Now I'm going to talk about the index structure for SAV. We use an R tree to query variants that overlap a genomic region. The begin and end positions of a variant indicated by the bars in this figure, the colored bars, are indexed with this tree structure. And this is similar to TABIX and CSI indices that are used for VCF and BCF. They also use an R tree, but there are some important differences, and I'm going to get to those in the following bullet points. The first difference is that we use a bottom-up layout. This allows us, when writing out a new SAV file, to simultaneously write out leaf nodes in the tree. And then once all of the records have been written, the internal nodes of the tree are generated and a footer is generated. We can then index to, or that we can then take this index that we've written out and append it to the end of the SAV file. This differs from previous approaches in VCF and BCF that store the indices as a separate file. So, with SAV, the index queries are done by first opening the file, then seeking to the end of the file, reading the footer, and then we can kind of read the index backwards in order to make those queries. And this footer also doubles as an end of file marker, which allows us to detect truncation during file transfers or whatever it may be. Another way that S1R is different is that it can be traversed on disk. That is to say, we don't need to load the entire index into memory, but instead we seek directly to the node, the tree nodes that satisfy our query. And so we start at the root node, and then from there we can determine which other nodes we need to traverse in order to finally get to the data that we care about. We also changed how we encode file positions in these indices. VCF, BCF, and SAV all split the file across compressed blocks, and with VCF and BCF, what is called they call a virtual offset is used to store the file positions of the beginning of the compressed block, in addition to the uncompressed offset within the block. And the problem with this is that it limits the size of the uncompressed data that can fit into a block to 4 kilobytes. With SAV, we instead store the number of records in the block as opposed to the uncompressed offset. And being able to store larger blocks of data improves the compression, especially with the Z-standard algorithm that SAV uses. And then there's also, in terms of the number of records being stored in the block, we can use that for other things I'm going to talk about, I think maybe in the next slide here. Yeah, so storing the number of records of the block allows us to do slice queries. And so this means we can scan the leaf nodes in the index and get the compressed block position of the nth record in the file, or a series of records with a start and end position. We can also quickly get the total number of records in a file just by looking at the leaf nodes of the index. And then if we wanted to evenly distribute work across compute nodes when parallelizing analysis of variance. So I just want to go over a quick history of this format. Our first iteration of SAV was a proprietary file format that only supported GT and HDS data types. So these are hard-coded genotypes and imputed haplotype, or basically imputed probabilities. And in addition to this limitation of what can be stored, there was another issue. So there have been many alternatives to BCF and VCF that have popped up in recent years, making it difficult for users when the format of their data isn't supported by the analysis tool they want to use. And so adding yet another alternative to the mix only adds to this problem. So for the second iteration of SAV, we decided to improve upon an existing standard instead of creating a brand new one from scratch. SAV version 2 extends BCF, which means that it gets all of the existing features of BCF and is easier to implement in software that already supports BCF. We're also a part of a GA4GH initiative that is discussing the future direction of the VCF spec and standard, and we hope to combine the best ideas from SAV with those of other formats in order to improve the standard. So I'm going to dive into the weeds just for a moment here to give you an idea of how we extended BCF. So the original BCF specification, in it, the data is encoded in what they call a typed value, which is basically a statically typed data vector. The type is encoded in the 4 least significant bits of the first byte, which is labeled as T's, as 4 T's in this diagram. But of the 16 possible values that can be stored in 4 bits, only 5 are currently being used for BCF, and that's the 5 entries on the table to the right. So what we've done with the version 2 of SAV is utilize some of this unused bit space to allow for a new sparse vector encoding, as well as a flag for PBWT. And so, certain vectors can either have this flag on or off to determine whether or not the transform is applied. And so by trying to make these minimal changes to the spec, the idea is that it's a lot easier to reuse the same code that already exists for parsing data, and some of the libraries that were designed to parse BCF, read and write BCF files, don't involve much changes in order to adopt SAV as an alternative format. Okay, so now I'm going to move on to some results. This table shows comparisons of deserialization speed on the top of the table, and then file size on the bottom. And so I think the main thing is that as sample sizes increase, the relative efficiency of SAV compared to BCF and VCF improves, both in terms of file size and speed. And I would say especially speed. I guess other thing to consider is that the PBWT improves size efficiency, but it does worsen the speed because of all that extra sorting that has to happen for each record. But we can enable it only for common variants. In other words, the least sparse vectors as kind of a compromise. So we can apply the sparse vector encoding for sparse vectors and use the PBWT for dense vectors. In order to evaluate whether the speed improvements of SAV would be noticeable when running a standard analysis, we ran a series of genome-wide association analyses and the results are in the table here. In the bottom half of the table, we're showing the results of doing these association tests using sparse vector operations, which results in a 50-fold runtime improvement on a 200,000 sample call set. And so even when you're not using the sparse vector operations, you can see a difference in these analyses, but you get the most bang for your buck when you when you can use sparse vector operations as part of your analysis, because you don't need to expand out that data into a dense vector. You can just leave it, compressed in memory as it exists on disk. Okay, so quick disclaimer here. This is a last-minute slide that I did this morning, so I hope there aren't any typos and that these numbers add up. But we're basically looking at runtime results of our Minimac for imputation software. And so we're looking at the total time it takes to write out the imputed results versus the total time of the analysis. And I have it done for both imputing 500 samples and then another run that imputed, looks like close to 8,000 samples. And there are a lot of things that go into the overall runtime. And so I wouldn't, consider this, extrapolate from here on what you would expect for other things. But the main point is that the using SAV as an output format does reduce the overall wall time compared to the other two. And I guess the other thing is that we use Minimac 4 as the backend to our imputation server or servers. We have two of them. And so we run, I don't know what the number is, but we've run probably imputed maybe many millions of genomes, and even a modest reduction of 10% or 20% of runtime, adds up in terms of compute costs. The other thing is that Minimac 4 is, the actual imputation can be parallelized, and so in these runs, I use 32 threads to do the analysis. And so if you're paying for a compute node that has 32 cores and you're doing single-threaded writing of the results to disk, then you're wasting a lot of CPUs when you're writing out the results files. And so shrinking that as much as possible is a more efficient use of those cores and the money spent on compute. So here I'm just giving some examples of using the command line interface. This is just, a small subset of all possible commands and options. But it does, pretty much all of the basic things. You can convert between file formats with import and export, and then also, subset on both dimensions with both of these commands, in addition to filtering out variance based on different criteria that exist in info fields. We can concatenate multiple files together, and we can also do this in a very efficient way that doesn't involve decompressing the files being concatenated together. We can just read the headers and make sure everything is compatible across the files, and then we can just do a byte for byte copy of the compressed data, which makes it a lot faster than it would be if we were to completely uncompress the data and then write it all back out again. We also have other common things like sorting the variants and looking at the headers and editing the headers. Also, they're designed to be very efficient. And then we can stat the file, but then we can also stat the index file. As I had stated earlier, this new index has some metrics that you can extract from it directly without having to read through the entire file. And so you can get things like, number of variants and number of variants with a certain chromosome that exist within the file. The next few slides, I'm just going to show you examples of the Savvy C++ API. It's pretty high level and hopefully easy for people to use. This is an example of just how you would copy a file from SAV format to BCF format. And it just involves initiating the input and output files and then looping through each variant and writing out. And that's, basically all that's involved. In this example, we're just showing some of the ways of accessing data when reading in a file. And so it's just really easy to get, very common information like the position of the variant, chromosome, the ref and alt. And then we can also get and set info fields and get and set the individual level data. Here's an example of how we do random access within the Savvy API. And this is just an example of doing region queries, but in a very similar fashion, you can do the slice queries that I talked about earlier. But you can basically just open a file and then call reset bounds as many times as you want to pull out different slices of variants within a given file. And then lastly, here's an example of subsetting samples. And so what we're doing here is requesting 3 samples indicated by their sample IDs in the requested variable, and then calling subset samples with it, and it returns the intersection between what you've requested and what exists in the file. And then from there on out, when you read in variants from that file, it's only going to give you data for that intersection. And yeah, it looks like I actually blew through these slides really quickly. So I hope I didn't glaze over topics here, but this is my last slide. I have just listed some of the tools that support SAV currently, with links to them. And final note, the last one in this list isn't released yet, and so it's still in a private repo, but hoping to get that out later this spring. But this tool is going to be an association analysis tool that takes advantage of all of the nice characteristics of SAV in order to speed up your analyses. So with that, I'm happy to take any questions.
Thank you, Jonathan, for that great presentation. If anyone does have questions, you can put them in the chat box or unmute yourself and ask the questions.
Hi, Jonathan. That was a great talk, by the way. I work at the Bioinformatics core, and so we're interacting from time to time with VCF or BCF style files. The API that you illustrated looked really clean, looked really clear. But as we're typically using tools that are off the shelf, what's the kind of recommended roadmap to integrate your file format with a tool that's optimized for a more traditional VCF style file format?
So one of the reasons that with the second iteration, we went with basically extending BCF was with the hopes of actually including it in HTSLib, which is the C API that not everyone uses, but I would argue many tools use HTSLib in order to access, or at least parse BCFs, because that's more of an involved file format. And while we haven't integrated it yet with that, I guess our hopes is that eventually it could more easily be integrated with HTSLib. At the very least, we might even make a fork of HTSLib that does this integration. And so that's one possible way. The other, that's probably, that when that's available, that will be the easiest way. But the other thing I want to put out is that while Savvy, the C++ library we developed, is cleaner, it's also faster. Sorry, one more slide back. So if you look at the top table here, I'm doing compute times for reading in BCF with HTSLib and then also with Savvy. And it's designed in a way to allow for more vectorized copying of data and transferring or altering of data. And so it's actually a bit more efficient as well. And so there are advantages of using the Savvy library, but I do take your point that there's a lot of existing tools and it'd be a headache to have to port all of them to a different backend.
Thank you. That's perfect. Thank you.
Great. So a couple of questions have come in via chat. So the first is from Dana, and she wrote, "Sorry if I missed this. Are location selection limited to chromosomes, or does this format allow for multiple areas of interest to be queried, such as all variants overlapping ChIP-seq peaks?"
All variants overlapping, what was that last part?
ChIP-seq peaks.
Yeah, I'm not familiar with that. And so my guess is the answer is no. The three ways of subsetting involve subsetting by genomic position within a chromosome, subsetting by samples, and then subsetting by the nth record within a file. So if you want to pull out the 500th record to the 900th record, you can do that really efficiently. But anything beyond that hasn't been implemented. No.
Great, perfect. And then the next set of questions came from Hari, who asked a couple of questions. Very nice presentation. One, am I right to say the highlight of SAV is space savings coming from BWT and speed from R-tree implementation? And then the second question, I'm happy to reread these if you want. I see SAV is already very fast, but I'm curious to learn how important parallel processing is in this domain and if there's a need to do so with SAV.
Okay, so for the first point, yeah, I would agree with that. I'd also point out that I think that in some cases, the speed of the file format is more important than the file size savings. And that's because, at least when you operate in a cloud environment, and you're running many analyses on a given data set, the cost of compute is going to far outweigh the cost of storage for those data sets. And so that's just kind of a side point I want to make. And then the second question, I'm sorry, can you repeat the second part of that again? I forgot.
Yeah, absolutely. So the second question was, I see SAV is already very fast, but I'm curious to learn how important parallel processing is in this domain and if there is a need to do so with SAV.
I see. So I guess the way I'm interpreting that question is that you're looking at, you're bringing up the possibility of doing parallel IO and doing parallel writes to disk. And there's usually an issue with parallel IO because the disk driver the disk hardware becomes the bottleneck, and it needs to do, at the end of the day, it can really, majority of the time, it can only seek and read one bit of, one sector of data at a time, or one block of data at a time. And so adding more parallel threads that are writing data to the disk at once usually doesn't benefit you that much. But one thing you can do with that is parallelize the compression. And I think that's more often what you see with these parallel IOs, that they're parallelizing the compression process and decompression process, which can be advantageous. That hasn't been implemented yet with Savvy, but that is on our radar and maybe in the future. Hopefully that addresses the question.
Great, thank you. Those are the only questions so far that have come in via chat. So if anyone has any other questions, feel free to put them in the chat box or unmute yourself.
I have a quick question, Jonathan. In your experience, does this set of libraries run pretty well on a workstation class machine, or do you want some with a little bit more horsepower to really get to the sweet spot?
I haven't done a thorough comparison of that, but it is going to play in. And so, there are two factors that are going to contribute to it. One is the CPU, the clock speed of the CPU you're using, and then also the data transfer speed you're working with. And so that might be over the network if you're using a network file system, or it might be just bare metal transfer speed, direct to disk. And so, both of those kind of play into it. And I think when you're comparing SAV versus BCF or VCF, in terms of the disk or the data transfer speed, the smaller file sizes is going to be the biggest component to, dealing with that. And so, regardless of how fast, well, I guess not regardless, if you have a really slow disk, SAV is going to be much better than VCF, or the discrepancy between SAV and BCF is going to be larger than if you're working with really fast disks. But then the other component is the decompression component. And in that case, if you have very fast CPUs versus very slow CPUs, which the discrepancy between those isn't as large, I think, between commodity hardware and HPC, you will see, you'll also see a pretty good improvement with slower CPUs, because there's less, especially when you're using sparse vector in-memory vectors, but even when you're not, because there's just fewer elements you have to iterate when you're pulling in these data vectors. So you don't have to iterate over all the zeros and copy them over to, from disk in memory. You can just copy over the non-zero values. So I don't know how much they're going to differ between. I don't have any empirical data as to like, how much it's going to differ between like your laptop or something on HPC, but I think in either environment, it's still going to perform pretty well relative to the other formats.
Thank you. That's perfect. Thank you.
Are there any other questions? We'll give it another couple minutes. Oh, it looks like another question did just come in to chat from Dana again. So she wrote, I think you touched on this in relation to the updates in SAV2, but do you have an estimate or way to track the uptake of Savvy format? The time saving seems very valuable, especially for large data sets, but curious if that's enough to overcome the activation energy for using something new.
Yeah, I don't have good metrics on that yet, and I agree that is one of the main challenges. And I guess my perspective is that I'm not necessarily I feel that issue. I feel that that difficulty of trying something new and all the effort involved in transitioning to something new. And so that's kind of why we're involved with the GA4GH initiative, and that we're trying to take what SAV offers, and then what other file formats in this field are offering, and trying to come up with a consensus of what a future standard would look like, so that we don't have this issue of people having to adopt these new formats as new things come out. So yeah, I mean, I don't think I have a really good answer for your question, but I do agree with the points that you're making.
So it looks like another question came in. Oh, no, never mind. No other question coming so far. So yeah, sorry about that. Just reading one that already came in. So yeah, if anyone else has questions, feel free to put those in the chat, or again, unmute yourself. We'll give it a few more minutes in case anyone thinks of any questions they want to ask.